Now in this part, let's talk about hemostasis. So hemostasis is a physiological mechanism that is going to eventually stop bleeding. And again, you don't want to confuse it with homeostasis. These are two totally different things. Hemostasis is a 3-step process, and it happens in this order. First is vascular spasm, where the blood vessels constrict. Second is the formation of a platelet plug, where the platelets are going to aggregate and collect together, stick together to plug the broken vessel. And then the third, and the one that leads to the permanent stoppage of bleeding, would be the formation of a blood clot, or the step of blood coagulation. So here we have step number 1, and that is vascular spasm. So what is it that stimulates these blood vessels to become spastic or to constrict? It is due to the injury. That is kind of an inherent response to injury in the blood vessels to constrict. We really don't know exactly what's going on, but we think it must be chemicals coming out of the endothelial cells or chemicals coming out of the platelets and other blood cells that are going to lead to vasoconstriction. It is a very short-lived response. At the maximum, it'll take about 30 minutes, and then that spasm will go away, and that's why we need to go to the platelet plug and then an actual blood clot to happen. But the vascular spasm, gives that time for the platelet plug to form. So step number 2 is the formation of a platelet plug. And I'm going to read this and then go on and explain it in a picture. So the platelet plug is going to start when there is a disruption of the endothelial lining. So remember that smooth endothelial lining prevented platelets from aggregating and collecting together. When you disrupt that smooth endothelium, you are now exposing the blood to the underlying collagen and other connective tissue, and they should not be exposed to those. And that's kind of like the stimulus for this whole process to start. The platelets are going to stick to a protein that are found in the found underneath the endothelium that is known as the von Willebrand factor. And then these platelets that just stuck to the endothelium are going to release chemicals from their vesicles. So remember platelets, when we talked about platelets, we mentioned that they have secretory vacuoles or have secretory vesicles that store different chemicals. We mentioned at the time serotonin, but there are other chemicals that are stored in there like ADP and thromboxane A2. Now the release of these chemicals from those platelets are going to activate them, and that process is known as platelet activation. Now these active platelets, through positive feedback, are going to bring in more platelets and stick to them. Okay, that process is known as platelet aggregation, and that keeps on going until we eventually make a platelet plug inside of the blood vessel. So we'll take a look at what on earth were we talking about. So this right here is an undamaged, this is a healthy endothelial cell with the subendothelium von Willebrand factor that you're seeing. This part is where the injury happened. So that exposed the von Willebrand factor, and that is going to bring in these platelets. Now these platelets are going to release their chemicals from their vacuoles, and those again were your serotonin and thromboxane A2. That leads to the activation of platelets, and now more platelets come in and they all stick together, okay, forming that platelet plug. This platelet plug can be very easily dislodged. It's another temporary solution, but it is one step towards stopping of bleeding. Now the platelets are aggregated in this area. Why is it that the platelet plug doesn't extend beyond the area of injury? And that is because the endothelial cells release chemicals like nitric oxide and prostacyclin. Prostacyclin is also known as prostaglandin I2. So nitric oxide and prostacyclin are going to inhibit and prevent platelets from aggregating in these non-injured areas. So you will find that the platelet plug will remain in the damaged part. Now for the permanent solution, which is a clot formation or blood coagulation, in order to make a blood clot, also known as a thrombus, a blood clot is basically fibrinogen that was circulating in our bloodstream that was a soluble protein. We have to change it into an insoluble protein known as fibrin. And it takes a long step, lots and lots of steps for that to happen. And that is because forming a blood clot is not something that we want to happen all the time. We are going to activate that system only when it's needed. So there are several steps in order to perform a blood clot. And there's actually two different sets of steps. So let us see. This right here is what a blood clot is. You can see now these fibrin proteins. These are now soluble, and they are tangling in all of the, you can see the red blood cells are tangled in there, and other blood cells are going to be tangled. So that's what basically a blood clot is. Fibrin tangling RBCs and other blood cells. So how is it that we activate this mechanism? Well, there's an extrinsic mechanism and an intrinsic mechanism. And extrinsic and intrinsic refers to the very first protein that turned on the switch for this whole process to happen. Okay, so all of these are different plasma proteins. And these plasma proteins are mostly made by the liver, and we're going to get to that in a minute. And when you activate one protein, that will lead to the activation of the next protein, which activates the next, and so on. So depending on the very first protein that you've activated, if that protein is found within the tissue, we call that the extrinsic clotting mechanism because it was not inside of the blood vessel. The first protein that you've activated in this cascade was a protein found outside of the tissue, outside of the blood vessels, and that is known as tissue thromboplastin. Okay, so we call that again the extrinsic clotting mechanism. But if the very first protein that you've activated in this whole cascade of events was found inside of the blood, namely Hagman factor, that is known as the intrinsic clotting mechanism. So either way, you have to activate the first protein that will activate the next, and so on. So again, if your first protein to be activated is outside of the blood vessels, outside of the tissue, that is known, namely tissue thromboplastin, that's your extrinsic clotting pathway. If you activate the plasma protein found in blood, known as Hagman factor, that is known as the intrinsic pathway. Either factors, again, are going to end up activating, different proteins until you end up with this common pathway. And, I'm not going to ask you to kind of remember these in detail, which ones are activated in detail, just the parts that are circled. So those are you are responsible for until we get to this common pathway, which is basically whether you started off as extrinsic or intrinsic, you're going to activate this prothrombin activator. Prothrombin activator is going to activate, as the name implies, it'll activate prothrombin into thrombin. Now, thrombin activates fibrinogen into fibrin. And remember, blood clots are fibrin, those strings that we saw, entangling RBCs. So that's how you're going to eventually make your fibrin clot. So that is your common pathway. A couple of things to keep in mind in these mechanisms. Either way, either pathways do need calcium, and we will see the importance of vitamin K as well. So clotting factors are produced by the liver. So all of these proteins that we have to activate in order to perform a clot are made or synthesized by the liver. Okay, they are secreted into the blood in an inactive form. And again, you do not really want them active in the bloodstream because that'll just keep on making blood clots. You want to keep them inactive until a blood clot is needed, and then you start to activate them in order. Remember that plasma without these clotting factors, we call that serum. Now, what is the role of liver in blood clotting? Okay, so we talked about how it's going to make those clotting factors, and in order to do so, it needs vitamin K. So what does the liver have to do with vitamin K? Vitamin K is one of those fat-soluble vitamins. Remember, liver makes bile, and bile is important to absorb, to digest and absorb fat solubles, to absorb fat and fat-soluble vitamins, including vitamin K. So in cases of liver failure, the patients that have liver failure have bleeding tendencies, and that is because they are no longer making bile, they are no longer absorbing vitamin K, which means that their liver cells can no longer make these clotting factors that we need. Now, what are the factors that oppose a clot formation? So having all of these clotting factors circulating in the bloodstream, even if they're inactive, what if somebody has atherosclerosis and that led to the activation of one of the cascades? What is it that our body can do to stop or oppose a clot from forming? Well, we have tissue factor pathway inhibitor. These are secreted by our endothelial cells, and that would prevent the activation of these plasma proteins. We can also deactivate thrombin, and that is going to be through this antithrombin 3. Remember, these are all just different proteins that we normally have in our bloodstream. Okay. We also have this thrombomodulin, and I'm going to go to the image to kind of explain how it works. So our endothelial cells have these receptors known as thrombomodulin. Thrombomodulin means that it's going to modulate thrombin, and that's exactly what these receptors do. They are going to attach to thrombin, modulate its configuration, and leading it to attach to another protein known as protein C. That will lead to the activation of protein C. So now protein C has become activated. This activated protein C deactivates factor 8 and factor 5. A stands for activated, which means, so if we kind of go back to the cascade, you can see here factor 8 and factors 5. These are two of the factors that we need on these cascades to make a blood clot. So that activated protein C can deactivate these two factors, preventing a blood clot from forming. Now, what if a clot is actually formed? Okay, so we do need clots to form. Now, how is it that we can get rid of the blood clot? We don't need that blood clot forever. So we will have to pretty much change that fibrin back into soluble fibrin fragments. And we do so by activating plasminogen into plasmin. And that would be by the action of these plasminogen activators that are found in our tissue. These are known as TPAs, or tissue plasminogen activators. Again, they activate plasminogen into plasmin, and plasmin is a protein that is pretty much going to break down that fibrin into soluble fibrin fragments, and that will eventually lead to the breakdown of the blood clot. So plasminogen activators, as we mentioned, the TPA, that is secreted by your endothelial cells. TPA is something that we make So it's a normal protein that our body makes, but it is found that if somebody has a blood clot, maybe they have a stroke or they've got a heart attack, or they have a blood clot maybe in the pulmonary arteries, if we inject these patients with TPA within 3 to 4 hours, that can lead to the dissolving of their blood clot. So these patients need to be identified as soon as possible, transported to a hospital. So if they arrive soon enough, again, within 3 to 4 hours of the formation of the blood clot, injecting them with TPA can dissolve that clot. Now, a couple of clotting disorders. So there's hemophilia, which is a genetic disorder. There is von Willebrand disease, where there's a reduced level of von Willebrand factor. We've seen how that factor was important for platelet aggregation. So in that disease, there's a decrease in the platelet plug formation. And then there's vitamin K deficiencies. So whether it's due to dietary deficiencies or due to liver failure, whatever the cause of the vitamin K deficiency is, that would lead to a decreased synthesis of the clotting factors by the liver, which leads to bleeding tendencies. And that is the end of chapter 12.